for sticking around for the last session. I'm going to present you an overview on Immatics Biotechnologies. We are a world leader in immuno-oncology with novel targets and in solid cancers. Immatics has developed two technology platforms, Expressed and Acceptor, for I/O target and T-cell receptor discovery, and based on novel targets that have not been used before in the space of immuno-oncology, we are translating this to deliver the power of T cells to cancer patients based on adoptive cell therapies and bispecifics. We have identified around 100 proprietary prioritized targets across 20 cancer indications with the goal to develop best-in-disease cancer therapies. The company has more than 150 employees based in Tübingen, Germany, and in Houston, Texas. We've raised more than $360 million in cash in venture capital rounds and upfront payments. And with this, we are building a proprietary and a partnered pipeline with 4 ongoing phase 1/2 clinical trials in our proprietary pipeline. This is an overview on the proprietary pipeline on the top, where we have 4 cell therapy trials in a unique joint venture with MD Anderson Cancer Center, currently recruiting patients and treating patients. We also have an allogenic platform that we're developing preclinically, as well as a bispecific platform that's also developed preclinically. In addition to our proprietary platform, we have a number of well-respected partners from the pharma industry. The 3 partnerships I'd like to highlight very briefly are Roche, which is an early-stage collaboration that we started in 2013, where we've reached the first successful milestone in 2017. Then 2 years ago with Amgen, a strategic collaboration to develop novel bispecifics, where we licensed 2 targets and programs to Amgen for an upfront of $30 million and up to $1 billion milestones and up to double-digit tier royalties. And our last collaboration that I will be actually talking about this afternoon, also in the private CEO forum, is with Genmab last year, where we have licensed 3 targets and programs to Genmab for an upfront of $54 million and up to $1.5 billion of potential milestones, and again, double-digit tier royalties. The basis for what our offering is that we have a world-leading and unique access to intracellular targets. You're probably familiar with the CAR-T space, immuno-oncology. CAR-Ts as well as antibodies are limited to surface targets only. So to tap into the much larger intracellular target space, which is 3 to 4 times larger than that surface proteome phase, you need to tap into the MHC presentation pathway. We've already heard about this also at the presentation by ISA, where vaccines are also going after such intracellular targets. And what differentiates us is that we've been working on discovery of such intracellular targets actually since the 90s academically, and since 2002 very systematically at Immatics. These targets are particularly helpful in tackling solid cancers, while CAR-T therapies have been highly successful in hematological indications, as you can see here. Actually, they have not been successful in solid cancers, but we do see with some first targets like developed by Adaptimmune and NY-ESO that they can be successful in solid cancers. We discovered these novel targets using our proprietary Expressed platform developed by Tony Weinschenk, our co-founder and chief technology officer. And this is an exercise we've been conducting for the last 15 years, very systematically. We actually started I/O target discovery when the word I/O didn't exist yet. What we use is at the core of the platform is high throughput, ultra high sensitive mass spectrometry, which we've built in-house. What we do is what's called quantitative immunopeptidomics, a term that has been coined by Immatics and Tony Weinschenk, where we measure the amount of peptide presented on the surface of tumor cells at a minuscule amount. So this is going down to femtoatomolar amounts, and compare that to the same peptide presented on normal tissue. And this is corroborated with further RNA-seq data, so that we have 2 independent data sets, a unique immunopeptidomics peptide mass spectrometry signal and a confirmatory RNA-seq signal. And that allows us really to go after real targets that are naturally presented. Many competitors do bioinformatics approaches where they actually predict peptides. That is a method that's highly error-prone. If you take any given antigen, like here, a tumor antigen that we've looked at, and you predict the top 50 peptides, even with the best algorithms around. And then you compare which of these peptides are actually truly presented on the surface of tumor tissues. You most often do not find the relevant peptides in the top 10 list. You see here that they're spread over the top 50 list, and actually in a very different way. So on top of confirming that these targets are real, that these peptides are presented, we can also describe how often do we find them and in what copy numbers. And that's absolute quantitation of peptides by mass spectrometry, something that only Immatics can master. So based on that, we have performed the human immunopeptidome program, a very systematic analysis across 20 different tumor types, 160 million peptide sequences eluted, and really boiled it down to 100 prioritized targets that are mostly focused on the allogenic type A2, but are now also extending this to many different other alleles. These are mainly 3 different buckets that we've created. Bucket number 1 are well-known and characterized parent proteins where we know the right peptide, like WT1. You know that a number of companies like Novartis, Juno, and Celgene are developing WT1 peptide-based cell therapies. Unfortunately, the peptide they are using is an artifact. It's never expressed on real AML tissue. We showed that years ago. This is also now confirmed by Celgene. They have discontinued despite being already in the clinic. So this shows how important it is to work with the real peptide and not with something that is not being confirmed by mass spectrometry. The second class are novel proteins like COL6A3 exon 6, where we have, which is a tumor-specific splicing variant that we've discovered. And the third bucket that we're currently working on are neoantigen-like peptides, cryptic peptides, but not those that are just found on a single individual patient, but are broadly shared actually among patients. So with these targets now, we are moving forward into 2 therapeutic applications, adoptive cell therapy and bispecifics, with adoptive cell therapy currently being the modality that's already in the clinic. Here we go. We have 4 clinical programs with 2 different therapeutic modalities, Actolog and ActEngine. Actolog is a personalized multi-target T-cell therapy, and ActEngine is classical TCR-T therapy, where we have an exogenous T-cell receptor that's introduced through genetic engineering and directed against one target per trial. Actolog, also called endogenous T-cells, is a technology that has been pioneered by Cassian Yi at MD Anderson, and MD Anderson has exclusively licensed the entire technology suite to Immatics. A patient comes in, we take a tumor biopsy, we measure 8 different targets in that tumor biopsy, and then apply in a very tailored fashion up to 4 different T-cell products for that particular patient, selected from the warehouse of these 8 pre-selected targets. So this is a tailored therapy where the 8 targets have been pre-selected, and it's a match and mix according to the specific presentation on that patient. It's also the first time that several, i.e. multiple defined T-cell products are given to a patient. We have started treating patients. We've so far treated 7 patients. The data for the first 3 patients we've published at ASCO. What the first finding that we've seen actually in the safety phase of the trial is that the biological efficacy that we've seen actually far surpassed our expectations. So while Castian Yi has seen typically levels of around 1 to 10% of the CD8 T cells ex vivo being reactive to the tumor antigens, we found this to be in an unprecedented level up to 50%. Actually, and this has not been observed before, so there's a lot of firepower that you can actually introduce by this Actolog cell therapy, because we're literally infusing billions of T cells per target into these patients. And we will soon also report some exciting clinical data that we are seeing in these patients. The other platform is ActEngine. This is classical TCR-T, so similar to what you've seen with Adaptimmune, Kite, and others, where we have a proprietary exogenous T-cell receptor that's introduced into autologous cells through genetic engineering. We have 3 different T-cell receptors against 3 different undisclosed targets, covering various different solid cancers, spanning from NSCLC, head and neck cancer, up to various gynecological cancers, with small trials up to 16 patients per trial, and upon clinical signal, then extending them. We're expecting to see initial clinical data actually in the second half of this year, and then headline data in the second half of 2020 for these trials. If you compare our targets, which are currently undisclosed, to the reference target in the field, NY-ESO, then our targets are obviously naturally presented as validated by mass spectrometry. They're similarly safe to NY-ESO, but they show considerably higher copy numbers than NY-ESO does, and a better prevalence in various different indications and tumor homogeneity, which is not shown here. So this is the advantage when introducing novel targets, that we can actually really develop best-in-class targets and therapies for these specific diseases. In Houston, Texas, we've built the entire competence to push forward such novel targets and T-cell receptors into the clinic, and covering the entire value chain. Have been very effective in actually from T-cell receptor to starting a clinical trial, doing this within 17 or 18 months, now shown and proven by 3 INDs that we've pushed to the clinic and where we are currently recruiting patients. It's very relevant for autologous therapies is the so-called vein-to-vein time, where our CMC group is actually innovating that process. Starting with 30 days for our first product, we're now down to 23 days, and we believe the commercial process that we're currently developing will be down to 13 days, which makes these autologous therapies competitive also. Beyond our clinical pipeline, we also are generating for cancer patients, our shareholders, very exciting future opportunities in our preclinical pipeline. Just briefly mentioning 2 of them, ACT-LO and bispecifics. ACT-LO is an allogenic T-cell platform, so not utilizing the autologous T-cells of the patient, but allogenic T-cells that have been taken from a different patient or donor, in this case gamma delta T-cells. We have a proprietary gamma delta T-cell expansion protocol that's unique because from one single leukapheresis packed from one donor, we can generate up to 9,000 doses, rather than 10 to 100, what you typically see now with companies like Allogene, Selectes, and others that have actually shown remarkable market caps and results in the clinic. We have tested this. This is still a preclinical development. Interestingly, despite that massive expansion, we do not observe any T-cell exhaustion, and we've successfully showed that these gamma delta T-cells can express T-cell receptor and CD8. The other platform that's currently developed for future opportunities preclinically is TCER, our bispecific platform. So bispecifics are essentially T-cell receptors, but instead of coupled or integrated in a cell, we're coupling them to a second specificity, again, CD3. So basically these bispecific molecules, similar to BiTEs that you've heard from Micromet, Amgen, other bispecifics, actually couple the tumor cell, covering or actually expressing that specific MHC peptide target that we've discovered to any type of T cell, and then recruiting and activating such T cells. In order to do that, we need T-cell receptors. We have a proprietary T-cell receptor platform called Acceptor that allows us not only to start with 100 to 150 T-cell receptor candidates per target, which is much more than our competitors like Immunocore do, but actually very diligently select those T-cell receptors that are truly not showing any on or off target toxicities, utilizing our unique XPresident antigen discovery platform. And then we stabilize such T-cell receptors, affinity maturate them more than 1,000-fold, retaining actually the specificity, which is really tough business to achieve, while sort of achieving that 1,000-fold maturation and incorporating that in a proprietary bispecific T-cell receptor platform with an extended half-life that's combined with antibody-like stability and manufacturability. We won't have time to dig into data, but this shows that we can retain the highly specific binding motif of the parental T-cell receptor that's already very clean with maturation, and that the proprietary bispecific T-cell receptor format that we're using is actually efficacious. So summarizing, Immatics, in our view, has revolutionized the identification of such tumor-associated PMHC targets through our XPresident technology. We have more than 100 prioritized targets and a world-leading T-cell receptor capability, a clinical pipeline of 4 adoptive cell therapies in solid cancers in conjunction with MD Anderson Cancer Center, a preclinical pipeline with ACT-LO and T-cell receptor bispecifics, as well as a number of interesting partnerships highlighting Amgen and Genmab, where we are expecting not just exciting milestone payments, but also new opportunities for cancer patients. With that, I'd like to close, and thank you very much for your attention.